The last two pieces of Cell Molec Part 1 deal with a couple of what are technically organelles that we need to know a little bit more about, and that is the cytosol and the cell membrane. So first here, Section 3 is about the cytosol. Now, in the past, you've probably heard more about cytoplasm than cytosol. Technically, cytoplasm refers to the interior of the cell. That includes the semi-fluid solution that all the organelles and cell parts are floating in, as well as the organelles themselves and the space inside of them. So basically, if you look at a cell, and then here's my nucleus, the cytoplasm is everything out here, including any structures or anything else that's in it. It's kind of really the college definition of cytoplasm. Now, in the past, you probably learned that cytoplasm was this semi-fluid stuff that's out here around all the organelles. Technically, that's referred to as cytosol. So notice the difference. Cytosol is primarily water with some stuff dissolved in it that fills the interior of the cell. Cytoplasm is the interior of the cell, minus the nucleus. Now, in the last 20 to 25 years, scientists have changed how they approach the cytosol. They used to think cytosol really was just sort of like this jelly that's floating in there. But today we know that the cytosol is really a very structured organization of proteins. And we call this the cytoskeleton. So just like you have a skeleton that is the framework of your body, the cell has a cytoskeleton that is really the framework of the cell. Now, the cytoskeleton has lots of different tasks. It's this huge network of proteins that does all kinds of different functions for the cell. The first thing we say is it is a dynamic scaffold for structural support. What does it mean if something is dynamic? It means that it's changeable. Now, like our building that we go to school in is not dynamic. If it was, I would not want to be standing in it because it would be changing and could fall down at any moment. But the structure of the cell, the cytoskeleton, is dynamic. It's changeable. It can change if the cell needs it to change. Now, it also serves as a framework that positions organelles everywhere that they're supposed to be. It acts as a highway system for transporting materials within the cell. There are a couple of proteins called dynein and kinesin. And these are motor proteins. These guys basically can drive along the cytoskeleton and move organelles. I'm just going to put move stuff inside cell. So these motor proteins, dynein and kinesin, can basically drive along the cytoskeletal proteins and move organelles or move other structures inside the cell. Cytoskeleton can also be involved in generating force, so if the cell needs to move, the cytoskeleton can help it do that. And then something that we'll look at later this year is the cytoskeleton is essential for cell division. So in order to do mitosis and divide, the cell needs some cytoskeletal changes to take place. Now, the cytoskeleton is made up of three major proteins or groups of proteins. These are all fibrous, so these proteins kind of have like a rope-like structure to them. And they are microtubules, which is the largest of these three. These are actually tubes made up of little proteins called tubulin. Intermediate filaments, the second one here, have that name because they are intermediate in size. So these are rope-like, they're very tough proteins that we see inside the cell. And then microfilaments are the smallest of these. And they are solid, thin structures composed of a protein called actin. So let's take a look at each one of these. First, here's a kind of a cool picture. This one on the right is just a little cartoon picture that appears in Dr. Wong and I's textbook, just kind of showing you how if you really look at that cytosol closely, you're actually going to see all these fibrous proteins in there. But then here on the left is the cool picture, which is a fluorescently tagged picture of some cells. So what they've done is they've tagged microtubules to show up green. As you can see, they form just sort of like this lattice work within the entire cell. They've tagged microfilaments, like actin, red. So you see you have sort of a network of those guys right underneath the cell membrane. And then a particular intermediate filament that is found in the nucleus is tagged blue. And so you see that glowing in that color as well. And you can really, in this picture, appreciate sort of this cobweb-like network of microtubules in particular. Okay, so microtubules. As we said, these are the largest cytoskeletal fiber. They are a big hollow tube that have tons of different functions for the cell. We'll kind of learn some of these functions as we move through the year, really, or we move through some more of our information about cells. But microtubules are essential for cell division. They're essential for movement of cells. Microtubules show a cool characteristic called dynamic instability. Remember, a minute ago we said dynamic means changeable or ever-changing. Ever-changing instability. So microtubules are constantly reorganizing their little tubulin building blocks, which is what this little guy is, to become shorter and longer. So the ability to constantly change their length allows them to help the cell do different things. So as this picture is showing you, they can be adding tubulin to either end. They can actually be taking away tubulin from either end as well, and it allows it to constantly get longer or shorter, whatever the cell needs it to do. As I said, there's lots of different functions here. Microtubules are a structural support network for the cytosol. They are those quote-unquote roads that serve as a transportation highway. So a minute ago, I talked about dynein and kinesin. Motor proteins like this can just walk along the microtubules to move structures within the cell. And then also tagged here in green in this picture are microtubules that make up the mitotic spindle, which hopefully you remember helps move your chromosomes around during mitosis, and we'll look at that more later this year. Now that second group of fibers in the cytoskeleton are the intermediate filaments. As I said before, these guys are intermediate in size, and that's why we call them intermediate filaments. There are a lot of different intermediate filaments out there. Depending on the type of cell you're looking at, you're going to find different types of intermediate filaments. So I don't expect you guys to know a whole lot about these, just the fact that they're there. They generally are just structural support. If you look at a nerve cell, you're going to see neurofilaments, which is a specific type of intermediate filament. These allow structure. Nerve cells, draw kind of a generic nerve cell up here. Nerve cells can be really long and thin, and so these neurofilaments will help provide structural support in these long parts of the cell so they don't just fall apart. Now, probably the most common or commonly known and most prevalent intermediate filament in our body is keratin. Keratin forms sort of a cage for cells that need to be really tough, and so you're going to see tons of keratin in your skin cells, which help make those parts of your body tough, and you're going to see more keratin in the parts of your skin that are tougher, so things like your palms of your hands and the soles of your feet are going to have more keratin. Keratin is also highly prevalent, that protein is highly prevalent in your fingernails and your hair. Alright, now the last group of filaments that we see as part of the cytoskeleton are the microfilaments. And microfilaments are the smallest group. Like we said, these are just little solid kind of strings made up of a little protein called actin. And actin is going to be found in really every cell, and it helps support the cell membrane and give the cell structure. But actin is also essential in your muscles. As microfilaments have this tendency to allow you to generate force. They might help a cell move, so a single cell that generates force, or they might help muscle cells generate force sort of in concert with other muscle cells, so that your entire big muscle on your arm or your leg or whatever can generate force to move your body. So the most common place to see actin is in the muscles, and they join there in the muscle with another protein called myosin to form these little units called sarcomeres. So when you look at a muscle, so let me draw a muscle here. This is kind of bad probably, but here's a muscle. You are just going to see at a microscopic level just thousands upon thousands of these sarcomeres. This is really getting ugly here. But basically, one of these little boxes might be zoomed in here, and you would see this little sarcomere, this little grouping of protein fibers. Now, what you'll notice is that the blue up here are the actin filaments, and the red things up here are called myosin filaments. Now, here's how this works. This would be a sarcomere that is relaxed. But when you go to flex your muscle, so think about making a muscle, and I know you guys have huge muscles, so you go flex your arm, and you got this big bulging muscle on your arm. When you do that, here's what happens. Right here, these little cross lines. The myosin can actually grab a hold of the actin, and then it's going to pull it toward the center. And the same thing is going to happen on this side. These little myosin red fibers are going to pull on these actin blue fibers and bring it together. So what you end up with is a structure that tightens, moving in from either side. So instead of having this gap like you see here, you might end up with something, let's see if I can kind of draw this, that is all closer together. So these blue guys are going to get close together and kind of be end to end here. And that's because the red guys pulled them closer together. Now think about that. When you flex your muscle, it kind of bulges, but it also gets shorter, right? And the reason that muscle gets shorter is because thousands of these things are lined up inside of there and they pull tighter. And all of that is thanks to this actin fiber working with myosin fibers. Alright, so that takes care of the cytoskeleton and the cytosol. The last thing we want to talk about here in part 1 of cell Molec is your membrane. Now, every cell that we know of has a membrane. I mean, if it didn't have a barrier around it, it wouldn't be a cell. So every cell, whether it's eukaryote, prokaryote, anything like that, is going to have a cell membrane. And that membrane is primarily made up of lipids, specifically called phospholipids, but there are also going to be proteins in there and carbohydrates in there as well. Now, a lot of times I just like to use the term cell membrane. A lot of different sources, textbooks call it a plasma membrane, but you can use either term. So the plasma membrane or cell membrane, pretty much the same thing. Now what I want to do in this section is just talk about the structure of that membrane. Then, when we move into Cell Molec Part 2 a couple weeks from now, we're going to start by looking at how the membrane works, because it's quite complex. You probably remember a little bit about diffusion and osmosis from your freshman biology class. That's what we're going to start with in Part 2. So here we're just going to look at the actual structure of the membrane, and then we'll worry more about how it works later. Alright, so phospholipids. Phospholipids are special lipids that have a glycerol backbone, or body, with 2 fatty acid chains that come off of that. Then they have a phosphate group attached to it, and they have what we oftentimes refer to as a head group. So the general way that we always draw these things is just like this, right? Now, the reason these are unique, and we've mentioned these kind of terms earlier this year, is because they have both hydrophilic and hydrophobic parts. So we call these amphipathic molecules. An amphipathic molecule is a molecule where part of it likes water, hydrophilic, and parts of it don't like water. Right? So hydrophilic likes water, hydrophobic does not like water. Now, when we look at a phospholipid, the head region is hydrophilic. It likes water. While the tails, or the tail region, are hydrophobic. Now, because of this, a membrane is formed by two layers of phospholipids. A double layer. We call this a bilayer. I think that word is actually coming up on the next slide, but we call this a bilayer, bi meaning two, and it's because you have water inside the cell and you have water outside the cell. So let's just draw a little line right here. Okay, so this is inside the cell, and that is basically a solution with water. And up here is outside the cell, and that also is basically a solution in water. So you're going to have these phospholipids, right? Remember, the heads like water. So the heads are going to orient themselves towards the water, and they're going to sort of hide the tails from that water. But the problem is, there's water down here too. So what happens is, another phospholipid lines up on this side, with its tails pointed that way. Now, if your cell membrane forms as a bilayer, a double layer here, then what we can effectively do is allow heads to always orient towards water, and tails, it's getting messy, to always kind of hide from the water because they're hydrophobic. And this layer of heads will block those tails from the water on the inside of the cell and the outside of the cell. So like I said, we call this a bilayer. It would look sort of like this, better than my little drawing, right? But then that's going to form as a sphere around the cell. So if you were to basically, you would never have these phospholipid tails exposed at all unless the cell gets kind of sliced in half, which is what we're looking at here on C, and then you'd be able to tell, okay, there's actually an internal layer and an external layer. Now, the phospholipids kind of float around, and there's cholesterol in there too that we learned about earlier this year, and there's proteins in there too. So there's cholesterol and proteins kind of floating around in this little sea of phospholipids. Now, if a protein is embedded in this membrane, we'll draw it kind of like this, we call that an integral membrane protein, right? It's integrated in the membrane. And most of these are going to serve as transporters so that things can get in or out of the cell. And so we'll return to that idea here when we get to the second part of cell Molec a couple weeks from now. Now this structure of the membrane leads us to the most kind of widely held theory about its structure, and we call that the fluid mosaic model. This is the idea that these phospholipids are present in a fluid state, right? So they're just kind of floating around, and they can move around laterally. So these phospholipids can move on one side of the membrane, but they don't usually flip from one side to the other, right? So the other piece of this, so that's the fluid piece. Mosaic just means made of lots of stuff, right? So mosaic, it is fluid because it is in a fluid state. It is mosaic because it's made up of lipids and proteins and carbs. Right? So all kinds of things put together. Now this illustration is showing you a famous experiment where scientists took a cell membrane. They artificially colored the outside of that membrane, so that's why this is pink on top. Then they used a laser to bleach away that color, right, to get rid of the color. So they ended up with a picture with an image like this. And they observed this under a really strong microscope. And what they saw is that those bleached phospholipids rather quickly started to spread out. They floated away until they were just kind of spread out all over that surface. So this is where scientists got this fluid mosaic idea. They were able to watch these phospholipids actually move around as though they were floating in this membrane. The way I like to think of this is if you took a pool of water and you dropped some ping pong balls in it, right? Those ping pong balls would float, and they would just kind of float and randomly spread out over time. And that's the way these phospholipids are sitting in the membrane. Now the very last thing is that while phospholipids move around a lot on one plane, right, one side of the membrane, they don't generally move from one side to the other, like from the inside to the outside or from the outside to the inside. That just doesn't work very well, and it requires energy to occur. So in order for a phospholipid to do this, it requires an enzyme called flipbase, and flipbase will actually use energy to move a phospholipid from one side to the next. Now scientists have suggested that this only happens maybe one time a month for a cell. So it's very rare that it needs to move phospholipids from one side to the other, and if it did, it would have to burn some energy to do that. Alright, this finally completes Cell Molec Part 1. Please let me know in class if you have questions. Otherwise, this should get you through your notes. Take care.